Hello everyone, hope all are doing good. So in the previous class, I was discussing lipids under what we discussed the introduction, classification, as well as the fatty acids, their definition, the classification of fatty acids into saturated and unsaturated. Even we discussed few of the examples that comes under saturated as well as unsaturated fatty acids. So today we shall start with glycerides. So what are glycerides? Glycerides are nothing but a kind of phospholipids. As per the definition, they are the esters formed from glycerol and fatty acids that are very hydrophobic. So in other terms, I can just simply define glycerides are those compounds which are obtained by the esterification of glycerol by fatty acids. In the sense, you can observe that glycerol has got 3 OH groups that are linked to carbon. The fatty acids, they replace this OH by oxygen and get attached to this glycerol. So, glycerides are obtained by the esterification of glycerol by fatty acids. Esterification in the sense the OH groups get replaced by oxygen and they in turn get attached to fatty acids. Glycerol has 3 hydroxyl functional groups which can be esterified with 1, 2, or 3 fatty acids to produce mono, di, as well as triglyceride. So here you can observe if a single fatty acid replaces one of the OH group, it produces monoglyceride. If it is producing if it is replacing 2 of the OH groups, then it produces diglyceride. If all the 3 OH groups get esterified by the fatty acids, then they are referred as triglycerides. So the triesters of glycerol with fatty acids are called triglycerides. You observed in the previous diagram, if all the 3 OH groups get esterified by fatty acids, then they are referred as triesters of glycerol, or simply you can define them as triglycerides. And the biological importance of triglycerides, which is much important. The first importance of triglyceride is acts as a source of energy. It's a major reserve source of energy, 9 kilocalories per gram in humans, plants, seeds, insects, birds, etc. In the sense, the triglycerides, they act as a source of energy. A gram of triglyceride can produce 9 kilocalories of energy. Second importance, they insulate the body against cold temperature, especially in aquatic mammals which live in cold region. We know that fats and oils, they act against cold temperatures. They provide warmness to the body. So in this way, they insulate the body against cold temperature. They help in maintaining the body temperature warmer. Third point, oxidation of triacyl glycerols yields metabolic water, reducing the need for drinking water in desert animals. Here, triglycerides also acts as water reservoir in case of desert animals. And the last point, as water repellent in some animals like whale. In the sense, these triglycerides that are found in the whale, they help in removing excess water from the water, or by avoiding the excess consumption of water, keeping whale in a balanced condition. Triglycerides can be of 2 types, simple triglycerides and mixed triglycerides. And the definition, triglycerides in which the 3 fatty acid radicals are of the same kind are known as simple triglycerides. If all the 3 fatty acids that are attached to the glycerol, if they are same, then those glycerides are referred as simple triglycerides. A mixed triglyceride is one in which the 3 fatty acid radicals are not all of the same kind. If all the 3 fatty acids that are attached to the glycerol are of different then they are referred as mixed triglycerides. Here you have the structure of a simple triglyceride, that is tristerine, and a mixed triglyceride. Here the structure that are written in green or indicated in green indicates the fatty acids, and if you just observe them, all the three are same. So it is a simple triglyceride. And if you observe the structure towards your right, you can see three different uh, fatty acids that are indicated by three different colors. It indicates that all the three fatty acids are different from each other, and this in turn yields a mixed triglyceride. Hope this is clear to you. Next, coming to the properties of triglyceride. First, we shall discuss the acid as well as the basic hydrolysis of triglyceride. We know that fats and oils are made up of triglycerides. Triglycerides contain three ester functional groups, because we know that glycerol has three OH groups, and three OH groups are in turn esterified by the fatty acids. So it in turn forms three ester functional group. Hydrolysis is a reaction with water. Triglycerides can be hydrolyzed, in the sense they react with water using different conditions. And here we have two different conditions of hydrolysis. One is acidic hydrolysis in acidic condition, and the second basic hydrolysis or alkaline hydrolysis, or generally referred as saponification in the basic condition. So we shall discuss acidic hydrolysis first. Acidic hydrolysis or acid hydrolysis of a triglyceride produces glycerol and fatty acids. Here you can see in this diagrammatic representation, triglyceride, it breaks down in the presence of three molecules of water into glycerol and a molecule of fatty acid. So this indicates acid hydrolysis of triglyceride, where a glyceride in the presence of water molecule or three water molecules break down into glycerol and a molecule of fatty acids. Next, we have basic hydrolysis, where this hydrolysis takes place in basic condition, in the sense in the presence of a strong base. Triglyceride breaks down into a molecule of glycerol and a soap in the presence of water as well as a strong base. So whenever the glyceride breaks down into glycerol and a fatty acid, it indicates acid hydrolysis. Whenever the glyceride breaks down into glycerol and the soap, that is sodium palmitate, then it indicates basic hydrolysis. Next, we have the concept referred as iodine number. The addition of iodine to unsaturated fats can be used to determine the number of carbon to carbon double bonds in the fat. The meaning of this sentence is that if you want to analyze if you want to determine the number of carbon-carbon double bonds present in a fat, then you can make use of iodine, which in turn helps in finding the unsaturation of the given fat. 1 mole of double bond reacts with 1 mole of iodine. Whenever you add iodine to any unsaturated compound containing double bonds, you should remember that a mole of iodine always reacts with a mole of double bond. A mole of double bond in the sense a single double bond is equal to a mole of double bond. Coming to definition of iodine number, the iodine number is defined as the number of grams of iodine that is reduced or that gets reacted with 100 gram of fat. I shall repeat, the iodine number can be expressed as the number of grams of iodine that is consumed by 100 gram of fat. Here we have a illustration or example to understand iodine number in a better way. So let me take one of the fatty acid, that is linoleic acid, whose molecular mass is 281 grams per mole with a formula CH3CH24CH double bond CH CH2CH double bond CH CH2 7 COOH. So this formula indicates the presence of two double bonds. And in the previous slide, I told you that 1 mole of double bond reacts with 1 mole of iodine, right? So 1 mole is nothing but a single double bond. A single double bond is equal to 1 mole of double bond. So here 1 mole of linoleic acid, that is with a molecular mass 281 gram per mole, it has got 2 double bonds. So I can easily tell 2 mole of double bonds. 1 mole of double bond reacts with 1 mole of iodine. So the mass of iodine is 254 gram. And here the linoleic acid, it has got 2 double bonds. So 2 mole of double bonds reacts with 2 mole of iodine, that is equal to 508 gram of iodine. So therefore 281 gram of linoleic acid, that is 1 mole of this acid, reacts with 508 gram of iodine. But as per the definition, iodine number is nothing but the mole of iodine, that is the grams of iodine that reacts with 100 gram of any given fat. So here I have to find the gram of iodine that is reacting with 100 gram of linoleic acid. So linoleic acid, 100 gram of linoleic acid reacts with 100 divided by 281, that is the molecular mass of linoleic acid, into 508 grams of iodine, which in turn gives you 181 gram of iodine. Therefore, the iodine number of linoleic acid is 181 gram. Hope I made it clear. Next, we have saponification number. You know that saponification is nothing but the process of preparation of soap. Excuse. And the definition, it is the milligram of potassium hydroxide required to saponify 1 gram of oil or fat. Mineral oils do not react with potassium hydroxide and are not saponified. So saponification number helps in finding whether the given oil is a mineral oil or a vegetable oil. Vegetable as well as animal oils have very high saponification value. Significance of saponification number. Each oil has got its own saponification number. Deviation from it indicates the extent of adulteration. Deviation from the saponification number indicates that some adulterant might be added to the oil. And the third point helps us to ascertain whether the oil reference is mineral or vegetable oil. As I said, mineral oil, they don't react with KOH and you don't get saponification number. If you don't get any saponification number, then you can easily determine that the given oil is not a vegetable oil, it might be a mineral oil. And the next property is rancidity of fats. Rancid means smelling odd, which is not an agreeable smell, a kind of awful smell. That is the meaning of rancid. Rancidity of fats and oils causes a disagreeable smell, texture, or appearance. So whenever you find any oil or fat smelling bad, we call it as rancidity. It's a property of any oil which undergoes oxidation, resulting in the formation of a rancid smell. This reaction occurs more quickly in the presence of heat and moisture, and this reaction is catalyzed by the enzyme lipase. This reaction is accelerated by the presence of enzyme lipase. The rancid smell is due to the release of fatty acids. Here you can observe in this schematic representation, I have a triacylglycerol which is undergoing oxidation in the presence of a catalyzed enzyme, that is lipase, which in turn breaks down into glycerol and a free fatty acid. Free fatty acid always gives some bad odor. That's the reason whenever the triacylglycerol undergoes oxidation in the presence of enzyme lipase, it breaks down into glycerol and free fatty acid, and we get this rancid smell. And this rancidity can be reduced by refrigeration. If you want to avoid the oil or the fats to get or to undergo rancidity, you better keep them in a refrigerated condition. In the sense, keep it in a cold condition. Yeah. Previously, I discussed the rancidity of fats, that is a hydraulic rancidity. Now I'm discussing oxidative rancidity or auto-oxidation. It occurs when oxygen is added across a carbon to carbon double bond. In case of hydrolytic rancidity, it undergoes in the presence of an enzyme referred as lipase, but here no enzyme is required. This is auto-oxidation, and this process, the rancidity takes place in the presence of oxygen, especially in the region of double bonds. Whenever the oxygen is added to the region of carbon-carbon double bonds, the fat or the oil undergoes rancidity. This reaction is catalyzed by light or enzymes and metal ions. It occurs in fats and oils with a high proportion of carbon-to-carbon double bonds. Can be controlled with antioxidants. The sites of reactivity are the carbon-to-carbon double bonds. Well, now I'm going to discuss about the structure and biological importance of some of the phosphoglycerides. So to start with, first we have lecithin. Lecithin is a phospholipid which consists of glycerol, 2 fatty acids, a phosphate group, and choline. And this lecithin, it is abundant in brain and nerve tissues. Therefore, it is gradually or generally used in treating the disorders concerned with memory issues like dementia and Alzheimer's disease. Alzheimer's disease is more common where a person loses his memory power by age. So lecithin can be used since it is found in brain and nerve tissues abundantly. It is used to treat Alzheimer's disease. Lecithin is taken as a medicine and is also used in the manufacture of medicines. A lecithin molecule in which both fatty acids are saturated aids oxygen uptake in the lungs. It's not only concerned with the memory issues, but it also helps in the uptake of oxygen by the lungs. And we have another species of lecithin where both the fatty acids are unsaturated, and this helps in the transport of cholesterol in the blood. So lecithin, as I said, it's a phospholipid which consists of glycerol, to which 2 fatty acids are attached, and to the third carbon a phosphate group, and which in turn attached to a choline. So if these 2 fatty acids are saturated in nature, they help in oxygen uptake in the lungs. If these 2 fatty acids are unsaturated in nature, they help in the transport of cholesterol in the blood. And the absence of lecithin, without lecithin and other membrane phospholipids, cells would be unable to maintain their structure and probably would dissolve back into their surroundings. So lecithin is much important in maintaining the structure of the phospholipid membranes. So this is about the definition of lecithin as well as its biological importance. Here we have the structure as well as the structural description of the lecithin. Lecithin, you can see the 3 carbon atom attached to hydrogen, they are the glycerol groups, and you have 2 fatty acids, C double bond OR, C double bond O R prime, and the third carbon of the glycerol is in turn attached to a phosphate group, which is in turn attached to a choline. So this is the structure of lecithin, which is also referred as phosphatidylcholine. Glycerol, which contains 3 carbon atoms, serves as the backbone of the lecithin molecule. 2 fatty acids are linked to glycerol at carbon atom 1 and 2. Here, you can just observe here the first carbon and second carbon are in turn attached to the fatty acids R and R prime. And the phosphate group is linked to carbon atom 3. Here, the one indicated with green. Choline is in turn linked to the phosphate group. Typically, though not always, the fatty acid attached to carbon 1 of glycerol is saturated, while that attached to carbon 2 is unsaturated. It depends on the phase shift of lecithin. They both might be saturated, they both can be unsaturated, or sometimes one can be saturated and other can be unsaturated. What matters is that the backbone should be of the glycerol, and the first and second carbon should be in turn attached to fatty acids, and the third carbon should be attached to a phosphate group, which in turn should be attached to a choline group. Hope I have made this clear. Next, phosphoglyceride, we have cephalene, is a phospholipid which consists of glycerol, 2 fatty acids, a phosphate group, and ethanol amide. The cephalene is somewhat similar to lecithin in its structure, because lecithin, it has got a glycerol backbone, and the first and second carbon are in turn attached to fatty acids, and the third carbon is attached to a phosphate group and a choline. But here, instead of choline, we have ethanol amine, a functional group. Coming to the biological significance, they have a role in blood coagulation. They accelerate blood clotting because they enter in the structure of thromboplastin. Thromboplastin is one of the component which is found in the blood responsible for the blood clotting purpose, which is essential for blood clotting. We know that whenever you get injury, your blood should clot within 3 to 5 minutes, or sometimes 6 minutes, not more than 6 minutes. If the blood keeps on flowing after 6 minutes, then you may have some anemic issues. So thromboplastin helps in blood clotting process. Coming to the structure, as I said, it has got a glycerol backbone. The first and second carbon are in turn attached to fatty acid, and third carbon is attached to a phosphate group, which is in turn attached to ethanol amine. So the name is phosphatidyl ethanol amine, or simply we can call it as cephalene. So typically, though not always, the fatty acid attached to carbon 1 of glycerol is saturated, while that attached to carbon 2 is unsaturated. So these factors do remain same between cephalene as well as lecithin. Next, we have phosphatidylserine. Again, it's a triglyceride, a fatty substance produced in the body that covers and protects every cell in the body and is involved in the clotting function of the body. Excuse. It is particularly important for the proper functioning of nerve cells within the brain, helping to transmit messages between them. It plays a vital role in cell cycle signaling, specifically in relation to apoptosis. Apoptosis, it's a programmed cell death, or simply it is a cellular suicide. Programmed cell death, this action occurs whenever your body cells get injured or they undergo any kind of mutation. There is one more term referred as necrosis. Necrosis and apoptosis are always used in a wrong manner. Apoptosis is a programmed cell death where the cells which are of no use, where the cell's DNA has undergone mutation, such cells will undergo cell death, where the cells will be packed into a garbage kind, and then it will be taken by the immune system or the immune cells. But in case of necrosis, the cells which have undergone any kind of injuries, they will undergo the death. So apoptosis and necrosis, though the functioning are same, but the manner is different. Apoptosis, referred as programmed cell death or cellular suicide, it takes place in cells whose DNA has undergone mutation or whose cells might be cancerous in nature. So those cells will undergo programmed cell death. Those cells will be packed in a kind of garbage and later will be transported to the immune system. But in case of necrosis, it's a cell death of those cells which have got some damages or injuries. It is a key pathway for viruses to enter cells via apoptotic mimicry. This is a structure. Again, the structure is somewhat similar to your cephalin as well as lecithin. You have a glycerol as a backbone, and you have two fatty acids which are in turn attached to first and second carbon. A third carbon is attached to a phosphate group which is in turn attached to a amino group referred as serine. So it is a phospholipid. It consists of two fatty acids attached in ester linkage to the first and second carbon of glycerol, and serine attached through a phosphodiester linkage to the third carbon of the glycerol. Well, I'll be winding today's session. Hope you understood these things. Please do sit and practice the structures. Till then, take care. See you in the next session. Bye.